All right, we left off here. We've gotten to the rules of complementarity. The rules of complementarity simply state that in every base pair, everything that's joining the two sides of DNA together, wherever there's an adenine on one side, there will be a thymine on the other, and wherever there's a cytosine on one side, there will be a guanine on the other, right? Now, a long time ago, when we were talking about different kinds of molecular bonds, I told you that the reason why there are there are four important kind of molecular bonds is to allow life to do what it needs to do. So let me remind you that the covalent bonds are super strong bonds, and covalent bonds hold this deoxyribose to that phosphate. Let's do it on this side. This phosphate to this deoxyribose to that phosphate to that deoxyribose. All of this that's holding the backbone together, phosphate sugar, that's all held together by covalent bonds, okay? As a matter of fact, it's covalent bonds that hold the thymine onto the deoxyribose or the adenine onto the deoxyribose. But, what is holding these two strands together? Hydrogen bonds. And remember, I told you that hydrogen bonds are used in life when we've got a place where a little bit of Velcro would be useful. And you will learn that DNA gets copied by kind of Velcroing the two sides apart. And then one side will be used to make a perfect copy of that side. And that'll be a new DNA. And then this side will be copied to make a new DNA, and it'll go to the other cell, right? Also, later on, you'll learn that parts get kind of opened the way Velcro would open. So stuff can happen, and then it gets closed up again. So hydrogen bonds hold the two strands together. Alrighty. Now, it was on the basis of this that a fellow named Chargaff, I think that's his last name, I did not know him, he had figured this out already. Professor Chargaff had learned, and he just knew that it was important, but in his experiments, he had learned that in any sample of DNA that he took, there would always be exactly the equal amounts of adenine and thymine to each other, and as much guanine as there was cytosine, always. And he knew it was important, but he didn't know why it was important. And then these people called Watson and Crick, they figured out this double helix, and Dr. Chargaff went, oh, that's why, right? Because once there was a double helix of DNA, he was like, oh, there's always an adenine, the same amount of thymines because they match each other. But poor Professor Chargaff, he did not get the Nobel Prize, but Watson and Crick got the Nobel Prize and endless accolades, and Dr. Chargaff gets mentioned in my lecture every semester, okay? He'd probably rather have the Nobel Prize. All righty, so we've spoken about DNA. Let's talk about RNA. You should notice that DNA and RNA, hey, they look a lot alike. I mean, except for the blue and the red. Well, actually, they do look a lot alike. But DNA nucleotides are different right there. Do you see that right there? There's an OH, oxygen, hydrogen, and over here, whoops, just an H, okay? That is why this sugar in blue is called deoxyribose because they deoxygened it, deoxyribose, and this one is just called ribose. If I ask you if there is ribose in DNA, you will say no. Now, I was not asking you if the letters ribose are in DNA, okay? I'm asking you if there's ribose. Ribose is a different sugar from deoxyribose. So in DNA, there is deoxyribose, no ribose, and in RNA, there is ribose, no deoxyribose, okay? But otherwise, DNA and RNA, they're made out of nucleotides, and nucleotides are a phosphate sugar and a base, okay? Now, bases. I told you that the DNA bases, adenine, guanine, and cytosine, those are the same, but that the thymine that's used by DNA, there's a substitute for it in RNA, and it's this one, the U, which stands for uracil. URACIL, uracil. Right? But other than that, there still are the same rules of complementarity. And by the way I remember the binding rules of complementarity is I remember the G always goes with the C because they're both round letters, right? And the A binds to the U if it's DNA, or the A binds to the T if it's DNA, okay? So whatever's left. One weird thing that I did that I just noticed is I used the G and the C and I put them together with 3 lines, 1, 2, 3. But the A and the U and the A and T, I put it with 2 lines. Why did I do that? Oh, by the way, over here in this drawing on the lower left, T and the A, 2 lines, G and the C, 3 lines, dotted lines. Why? Because the guanine and the adenine are joined to each other by 2 hydrogen bonds, whereas the guanine and the cytosine are joined together by 3 hydrogen bonds. Remember, I was telling you that just like black, white, red, yellow, these 4 nucleotides, either the 4 for DNA or the 4 for RNA, they are as different from each other as black, white, red, yellow from the point of view of the little machines that have to build DNA or build RNA. And one of the ways they're different is in the size of the molecule. Another way that they're different is in the number of hydrogen bonds that they're able to form. And so if you try to put a G with a U, they just don't fit together, partly because of these hydrogen bonds. Okay. Now, DNA. We know what DNA. We know what DNA is. We know what DNA is, but now we need to talk a little bit about RNA. So DNA, that's what your genes are made of. That's what your chromosomes are made out of. Those are the instructions to how to build a perfect U, and that stuff is kept in the nucleus because it's precious and we don't want it to be damaged. What is RNA? RNA is also considered genetic material, but RNA is mostly expendable stuff. DNA is super valuable. Those are like the original blueprints. RNA is going to make up either the machinery that builds the protein or machines that help with building the protein, or like a temporary Xerox of the gene so that the protein can be built, okay? And the 3 kinds of RNA are messenger RNA. The M in mRNA stands for messenger. I don't think you can read that. I'm sorry. Messenger. The second one, ribosomal RNA, that R stands for ribosomal RNA. And the T in front of the last one stands for transfer. Okay? We're going to talk about them in detail. So the 3 kinds of RNAs that you need to know for this module, which is all on exam 1, is mRNA, rRNA, and tRNA. All righty. Now, why is there a genetic code? Well, let's review what we've learned so far. What we've learned so far is that DNA is made out of G, A, T, and C. We won't mention RNA for a moment. And yet, I've told you a few times now that this stuff called DNA, that this is what it holds the instructions for how to make all of your proteins. Well, that's a problem, isn't it? Because there are 20 different amino acids, but there are only 4 different nucleotides. So how can a string of G, A, T, and C, how can they code for all of my amino acids? And the answer is that there's actually a genetic code. Okay. By the way, there's an old movie. There's an old movie called Gattaca. You guys are sitting around at home. You may be interested in an old movie. It's called Gattaca. Gattaca. And Gattaca stars Jude Law, Uma Thurman, and Ethan Hawke when they were young and adorable. They were like your age in this movie, okay? And the movie's called Gattaca because it was about a science fiction future where your DNA is your destiny. If you want to get into medical school, they take a sample of your DNA and give you a thumbs up or a thumbs down, okay? So Gattaca. By the way, Gattaca is also how I remember which of the nucleotides are DNA nucleotides and which of them are RNA nucleotides, because Gattaca is made out of G's and A's and T's and C's, and that's the name of the movie, all right? So why do we have a genetic code? We have a genetic code, first off, because if we've just got four things like black, white, red, yellow, and they always match black, white, red, yellow, then it is really easy to make copies of these strands with great fidelity. Fidelity is the word that they use. They mean no mistakes. There's much, much less than 1 in a million mistakes when it comes to copying your DNA. Okay? Now, the problem is that if you had just one nucleotide for every amino acid, then you wouldn't be able to code for 20 different amino acids. You would have to code for only 4 amino acids, and that's not going to work. We've got 20 amino acids, and we've got only 4 nucleotides. So we use a genetic code, and we do that with things called codons. All right, so let's just imagine for a moment. Let's imagine that I was going to give you an A as long as you could use these symbols to translate the first paragraph of my syllabus, okay? And if you did this and you were like, okay, I'll use that for an A and that for a B and that for a C and that for a D, that's not going to work, right? Because there are more letters than that in the first paragraph of my syllabus. But you could do it, right? But you'd have to say, okay, for A, I'm going to use square, star, star. That's going to be A. And for the letter B, I'm going to use square, square, star. And for the letter C, I'm going to use square, square, square. And for the letter D, I'm going to use star, star, star. Stop me before I go any further, right? What I'm trying to explain to you here is that by using, you would have to create a code. When we talk about the genetic code in life, we're not talking about it poetically. We're talking about it literally. There is a literal genetic code, and it took us a while to figure it out. But now that we know, we can look at the DNA that looks like Gattaca, and we can figure out exactly which amino acids in a row will be created with that gene to make a protein, okay? So a codon is a term for 3 nucleotides in a row. That's like a little code word for one amino acid, right? We're going to start there at the beginning of our next lecture.